Good morning. Today we have to study fruit in 12th class biology in the sexual reproduction of flowering plants. We know that fruit is the product of the ovary. Fruit formation takes place from the ovary. Such type of fruit is called true fruits. And in some cases like the strawberry, like in the apple and the cashew, fruit formation take place from the thalamus, from the receptacle. Such type of fruits are called false fruits. You can see the figure. So in this figure it has been shown that these two types of fruits are here, and you are clearly looking that seed is inside the in the center of the fruit, and around them seed coat is there, endocarp or mesocarp has been shown, and the thalamus has played important role in the fruit formation. This is thalamus, and in this figure this is thalamus. So this thalamus has played role into the fruit formation. Second thing we know that, as we know that fruit formation take place after the fertilization, but in some cases without fertilization fruit formation take place. And we know that if the without fertilization fruit formation taking place, that fruit becomes seedless. Such type of fruits which are the results of syngamy, which are the result of any syngamy. No syngamy occur, no fertilization occur, and the fruit formation is taking place. Such type of fruits are called parthenocarpic fruit. And this parthenocarpic fruit are seedless. And in this parthenocarpic fruit we can obtain banana and we can obtain grapes. We have to know that the ovary wall develops into pericarp, and this pericarp is the very tough seed coat, and this tough seed coat protects the seed. The state of inactivity of the metabolic rate in the seed, which is caused due to the 10 to 15% remaining moisture of the total mass of the seed in the seed, is known as dormancy. Dormancy is the state of inactivity of the metabolic rate of the seed. How does dormancy cause? Then you can understand, when the moisture quantity becomes 10 to 15% of the total mass of the seed, then dormancy is caused, then metabolic rate greatly slows down. We have to know that these seeds will be dusted, will be utilized by different organisms, by human beings too. And the next year, out of these seeds we can cultivate, we can use some seeds into the cultivation, into the agriculture, and in the creation of new individuals of the plants. But we have to know that what is the viability of all these seeds. The different types of plants are making different types of seeds. What is the viability? Till what time this type of seeds can germinate, can survive, and have germination capacity? A record, a plant we have obtained, a seed we have obtained. That seed was dispersed 10,000 years ago from that parent plant. And its plant's name is lupin, Lupin arcticus. Lupin arcticus. And this Lupin arcticus, Lupinus arcticus, is the plant whose seed was dispersed 10,000 years back from their parent plant. And that was in the dormancy. And this plant, this seed has germinated 10,000 years later from their germination day, germination time. This is the record viability period of any seed. Second record we have obtained from the date palm. The seed of this date palm was dispersed 2000 years back. And after the dispersal from their parent plant, this seed 2000 years later, this seed has germinated. And this plant is date palm, and scientific name is Phoenix dactylifera. Phoenix dactylifera. We have to know that. We know that fertilization leads and form the seed. Ovule forms the seed. But in some species of Asteracean grasses, it has been seen that without sexual reproduction, seed formation take place. without sexual reproduction fruit formation take place. This process of seed formation, this process of formation of such type of plants, such type of flowers have fruits having the seeds is called apomixis. Generally we can say that apomixis is the mode of asexual reproduction that mimics sexual mode of reproduction and forms seed. In apomixis, we have to understand that there is not haploid egg is formed. There is not fertilization is taking place. In this case, we can see that nucellus, which is found around the embryo sac, that nucellar cells protrudes inwards, come into the embryo sac, and develops and form the embryo and leads to the seed formation. We can imagine there are many nucellar cells are formed around the embryo sac. You can see this figure. This is the embryo sac, and around this many nucellar cells are formed. This is the embryo sac, and around this many nucellar cells are formed. And these nucellar cells protrude inward, come into the embryo sac, and develop and form the embryo. And as many nucellar cells are formed, many embryos are formed. The process of formation of many embryo from many nucellar cells is known as polyembryony. The process of finding of many embryo inside the embryo sac or ovule is known as polyembryony. In mango and citrus, it has been seen that egg formation takes place, but that egg is diploid. There is not reduction division in the egg. There is not reduction division in the egg, and that egg is diploid, and that diploid egg develop and form the embryo. That diploid egg develop and form the embryo, and that embryo leads to the seed formation. In some cases, it has been seen that embryo sac in seed breeding industry, we know that artificial hybridization is done. And after the artificial hybridization, they select the two good variety breeds, two good quality crops, having the desired set of characters. And these two good quality crops are crossed. And after crossing, the new hybrid seeds are obtained. And these new hybrid seeds are cultivated and have the desired set of character. But it has been suggested that apomixis, which is the process in which the sexual reproduction is mimicked, and that is actually the asexual reproduction, there is not reduction division. If the reduction division is not happening, then there is not segregation of the chromosomal set. And if the chromosomal sets are not segregated, then this can carry the set of character from generation to generation. Actually, in the seed breeding program, what happened? When the two haploid set of characters are allowed to fuse, fertilize, and make the hybrid seed, and farmers again after the hybridization, when the plant and these plants are making the hybrid fruits are utilized, and some of their seeds again cultivated next year, then it has been observed that in the next year the seeds do not carry the characters. Because during the stamen and pistil formation, during the male gamete and female gamete formation, meiosis occurs, and meiosis results into the segregation of the set of characters. And this set of characters was accomplished after the artificial hybridization, and this was the costly breeding program. So in each and every generation, during the formation of male and female gamete, if the set of chromosomes or characters are segregated, then in the next year those set of characters will not be carried, and whatever the output the farmer want they will not be in the position to obtain that productivity, because the set of character have been segregated, have been separated, have not been maintained. But if the apomixis process, which is the asexual mode of reproduction, if the apomixis process is used, the seeds will be formed because the diploid egg is developing, because the diploid nucellar cells are making embryo. Then mitotic division will be taking place in the diploid egg in the nucellular cells. So there is not segregation as meiosis is not going on. So the set of characters, the clone of characters will be carried generation to generation, and these seeds which will be formed, which will be the output of the productivity of the crop, will not be lost, and our productivity will be constant. Thank you.